Hello, ma'am. Good evening. My name is Dharya Trivedi.
And my name is Vidya.
Today we are doing presentation on Helminths in detail. So let's start on understanding Helminths in detail. So let's start. Introduction to Helminths. Helminths are parasitic worms that can infect humans and other animals. They belong to the phylum Platyhelminthes, also called as flatworms and nematoda roundworms. Helminths can be free-living or parasitic, and they play a significant role in shaping ecosystem and impacting human health. Uh, Definition of helminths. Helminthiasis refers to an infection caused by helminths. When these worms enter the human body, they can cause parasitic infections. There are three main types of helminths. First, roundworms or nematodes. Second, trematodes or fluke. Third, cestodes or tapeworms. Nematodes are cylindrical worms that can infect the intestines or other parts of the body. Trematodes are flatworms with leaf-shaped heads that generally infect bile ducts, liver, or blood. Statosaurs, long segmented flatworms found in or around the intestines. Importance of studying helminths. Understanding helminths is crucial for healthcare professionals, diagnosing and treating helminth infection, researchers studying their biology, distribution, and impact on ecosystem. Third, general awareness, recognizing the role of helminths in a global health and disease. Classification of helminths. Helminths are broadly categorized into three main groups. Nematodes, these are cylindrical worms with a tubular body. They include both free-living and parasitic species. The examples of nematodes include Ascaris lubricoides, also known as intestinal roundworm, and Wucheria bancrofti, which causing lymphatic filariasis. Second, cestodes. Tapeworms are flat, segmented worms that live in the intestines of vertebrates. They lack a digestive system and absorb nutrients through their body surface. The examples include Taenia solium, also called as spoke tapeworm, and Diphyllobothrium latium, also called as fish tapeworm. Third, trematodes or flukes. Flukes are leaf-shaped flatworms that parasiticize various organs such as liver, lungs, and blood vessels. They have suckers for attachment. The examples are Schistosoma mansoni that causes schistosomiasis, and Fasciola hepatica causes liver fluke. The general characteristic of helminths are the helminths exhibit the following features. The complex life cycles. Helminths often have multiple life stages, example larva and adult, and may require intermediate hosts. The parasitic lifestyle. Most helminths are parasitic, relying on a host for survival and reproduction. Third, diverse morphology. Helminths vary in shape, size, and structure. Their impact on health. Helminth infections can cause diseases in humans and animals. Their global distribution. Helminths are found worldwide, affecting millions of people. So, nematodes, also called as roundworms. The example of nematodes are Ascaris. These are also known as the intestinal roundworm. Ascaris lumbricoides is the one of the most common human parasites. It resides in the intestines and can cause ascariasis. Second, hookworms. Hookworms, also called as Nectar americanus and Xylostoma duodenale, that attach to the intestinal lining and feed on blood. They cause hookworm disease. Third, pinworm. Enterobois vermicularis is responsible for pinworm infections characterized by itching around the anus. Their general features. Morphology. Nematodes have a cylindrical body shape with a tough cuticle. They lack a circulatory system. Second, life cycle. Nematodes typically have a direct life cycle involving eggs passed in feces larval stage, and adult worms. Third, habitat. They inhabit soil, water, and various host organisms. Disease impact. Nematode infections can lead to gastrointestinal, pulmonary, or skin related disease. So let's see the images of nematode or roundworm. These are the Trichinaria spiraliasis, second Enterobis vermicularis, and third is Enclastosoma duodenale and Nectar americanus. Cestodes or tapeworms. Cestodes are parasitic flatworms with long ribbon-like bodies that can grow over 20 meters in length, depending on the species. Their bodies are segmented, which plays a crucial role in their life cycle. Some common cestodes include Taenia solium, also known as pork tapeworm, Taenia saginata, the beef tapeworm, Echinoconus granulosus, and Echinococcus multicochlearis. This cause haploid disease. Diphyllobothrium latium, the fish or broad tapeworm. Hymophis nana, the dwarf tapeworm. Spirometra spp, the pleriosericoid tapeworm larvae, causing sphagnoses. The morphology. Taenia saginata. They usually have less than 5 meters long, can grow up to 25 meters. Pear-shaped haired scolex with 4 suckers, no hooks. Long flat body with hundreds of segments called proglottoids. Egg producing sections. Eggs are round and yellow brown. The Taenia solium. Variable size up to 7 meters. Globular scolex with 4 suckers and hooks. Regulatoids are 5 into 10 mm with branching trees. Their life cycle. Humans ingest poorly cooked meat containing tapeworm larval cysts, sister cali. The larvae attach to the small intestine by the scolex suckers. Over 3 to 4 months, the tapeworm matures and proglottoids develop. The gravid proglottoids release eggs into the feces. The excreted tapeworm eggs survive on vegetation and are consumed by cattle or pigs. In these animals, eggs hatch releasing cysticercai. Humans get infected by eating raw meat with cysticercus. Cestodes. The platyhelminthes or flatworms showing clonorchis, and in second image it's Taenia sp. Trematodes. General feature of trematodes or flukes. Trematodes are parasitic flatworms that infect various organs in their host. They are often categorized based on the principal organ system they invade, the liver and bile duct. Example includes Clonorchis sinensis, Fasciola hepatica, and Opisthosis species. The lumen of gastrointestinal tract. Example includes Fasciolopsis busci, heterophyes, and related organisms. Lungs. Examples: Paragonimus westermanni and related species. Uh, vascular vasculature of a gastrointestinal or urinary, urinary system. Example: Cystosoma species. Their morphology. Trematodes are dorso-ventrally flattened, often leaf-shaped. They possess oral and ventral suckers. Most are hermaphroditic except for cystosomes. Their eggs are operculated except for cystosome eggs. Their life cycle. Trematodes have complex life cycle involving intermediate host, often snails. Humans can become infected by ingesting contaminated water or undercooked fish or plants containing fluke larvae. The larvae develop into adult flukes in the host, where they reproduce and release eggs. The trematodes. The image showing the liver fluke, where its a size is 3 mm, showing the ovary, oral sucker, ventral sucker, u- uterus, and cecum. External structure of helminths. Cuticle and teguments. Both nematodes and trematodes have a protective outer covering. The cuticle, non-cellular collagenous layer, provides structural support and protection. Molting occurs to allow growth. Second, tegument, central layer covering the body, function in nutrient absorption, waste excretion, and protection. Contains microvilli for increased surface area. External structure of helminths. So this showing rostellum, hooks, and suckers, which together also known as colex. The long nail and the immature proglottis. And this is the enlarged view showing cuticle and gravid proglide with eggs. Nematode external structures. The cuticle. The cuticle is the outermost layer of the nematode's body wall. It serves several essential functions. The protection. It shields the nematode from the harsh external environment, including predators, pathogens, and destination. Exoskeleton. The cuticle acts as an exoskeleton, providing structural support and maintaining the nematode's shape. Movement. It facilitates movement through soil and plant tissue. The cuticle consists of multiple layers and is made of proteins. It may exhibit striations, annulation, and punctuations. Specialized structures such as sensory papillae or setae may be present on the cuticle. The alae, wing-like structures. Alae are lateral extensions found in some nematodes. They resemble wings and are located along the body, typically in the cervical, head, and tail regions. These structures play a role in locomotion and may aid in burrowing through soil or host tissue. Alae are particularly noticeable in certain nematode species. The papillae. The papillae are sensory structures found in nematodes' head, cephalic region. They can be spine-like or finger-like. These papillae help the nematode detect environmental cues, such as chemical signals or mechanical changes. Papillae contribute to the nematode's ability to navigate its surroundings and locate suitable hosts or food sources. The cestode's external structure, the scolex. The scolex is the anterior end of the tapeworm. It serves as an attachment organ, allowing the tapeworm to anchor itself to the host intestinal wall. The scolex typically has suckers, which used for attachment, hooks present in some species. Example, Taenia soli. Think of the scolex as the tapeworm's heads. The proglottids. The proglottids. Behind the scolex, the tapeworm body consists of a chain of segments called proglottids. Each proglottid is a reproductive unit. As the tapeworm grows, new proglottids form at the posterior end. Mature proglottids contain male and female reproductive organs. Gravid, egg-filled proglottids release eggs into the host species. Trematode external structure. The tegument. The tegument is the outer covering of the trematode's body. It is a living layer that allows the absorption of nutrients. Think of it as a protective skin that surrounds the entire body. The oral sucker, located at the anterior end of the trematode, surrounds the mouth, used for feeding and attachment to the host, and imagine it as a suction cup near the mouth. The ventral sucker, also called as acetabulum, found posterior to the oral sucker, also used for attachment to the host, helps the trematode maintain its position. Picture it as another suction cup on the underside of the body. So thank you. Now I would like to call my friend Rich for the further information on helminths. Thank you.
Thank you, Daria, for passing the information. So let's start with the life cycle overview. So life cycle overview. Let's start with the trematodes or flukes have complex life cycles that involve both definitive and intermediate growth. They typically have an egg stage, miracidium, or free-swimming larva, sporocyst, rhidia, cercaria, and metacercaria. The metacercaria are usually ingested by the definitive host through contaminated water or food. General life cycle stages. Eggs. Trematode eggs are released into the environment through the host species. Miracidium. The miracidium hatches from the egg six times suitable behavior. These larvae Cercaria emerge from the snail and swimming freely in water. Metacercaria insist on vegetation on or other surfaces, waiting to be ingested by the definitive host. Differences among nematodes, cestodes, and trematodes. Nematodes, or roundworms, long cylindrical bodies with a complete digestive system. Separate sexes, male and female. Common examples are ascaris, hookworms, and pinworms. Cestodes, or tapeworms, flat segmented body with no digestive system. Hermaphroditic, both male and female reproductive organs attached to the host intestine using suckers and hooks. Examples are tinea, beef tapeworm, fork tapeworm. Trematodes or flukes, leaf-shaped bodies with oral and ventral suckers. Hermaphroditic examples, cystoma, fasciella hepatica, liver fluke. Nematode life cycle. So first you are starting with the egg stage in which the adult female Ascaris lumbricoides produces approximately 22 lakh eggs per day. These eggs are passed with the feces into the environment. Unfertilized eggs may be ingested but are not infected. Larval development. Within fertile eggs, larvae develop to infectivity. This process takes 18 days to several weeks depending on environmental conditions. Optimum moist, warm, shaded soil. Swallowed eggs. After infective eggs are swallowed, the larvae hatch in the small intestine. They invade the intestinal mucosa and are carried via the portal circulation to the lungs. Lung phase. In the lungs, larvae mature further over 10 to 14 days. They penetrate the alveolar walls, ascend the bronchial tree, bridge the throat. Swallowed again. Larvae are then swallowed again. Upon reaching the small intestine, they develop into adult worms. Adult worms. Adult worms live in the lumen of the small intestine. The entire life cycle from ingestion of infective eggs to oviposition by adult female takes 2 to 3 months. Adult worms can live up to 1 to 2 years. So let's take a look. Here, as you can see, there's a ship and there are three living stages. First, the eggs and dung from the ship, then it goes to first stage larva, then it goes to second stage larva, then the third stage infective larva from 2 plus 12 weeks, that is 14 weeks, and then it again goes to the ship, which is the L4 stage. Let's go to the second. Egg stage, young infected with Cliniasolium past egg or gravid probabilities in the species. These eggs can survive for days to months in the environment. The intermediate host, pigs become infected by ingesting vegetation contaminated with eggs or gravid proglatids. In the pig's intestine, the oncospheres, heads from eggs, invade the intestinal wall and migrate to the striated muscles. There they develop into cysticerci or encysted larvae, which can survive for several years. Human infection. Now, humans become infected by ingesting raw or undercooked infected pork containing cysticerci. At the human intestine, the cysticercus develops over 2 months into an adult tapeworm. The adult tapeworm attaches to the small intestine by its pollex and resides there. Now, it becomes adult tapeworm. The length of adult Linear tapeworms is usually 2 to 7 meters. They produce proglottids that mature, become gravid, detach from the tapeworm and migrate to the anus or are passed in the stool, approximately 6 per day. Each gravid proglottid may release up to 50,000 eggs. This is the life cycle of the sex toad. So you can see in the picture. First, it goes the human primary host. It's undercooked pork containing tapeworm cyst. Then the larva emerge from cyst and attach to intestinal wall. Then the tapeworm matures in intestine and produces a long series of reproductive segments, each containing thousands of eggs. Then proglottids are shed from end of worm and are passed into feces, as I told earlier. Now, the pig, which is the intermediate host, is food contaminated by infected feces. Larvae hatch in pig intestine and migrate via blood vessels to muscle tissue, and then the larva from cyst in muscle tissue. So the life cycle continues. Now, the trematode life cycle. Egg stage. Humans infected with cystoma pass eggs in their feces or urine. These eggs enter freshwater environment. Under suitable conditions, the eggs hatch, releasing Miracidia, free-swimming larvae. Snail intermediate host. Miracidia swim and penetrate specific freshwater snails. Within the snail, Miracidia develop into sporocysts. Sporocysts give rise to a second generation of sporocysts and eventually form Cercaria. Cercaria release. Cercaria with forked tails leave the snail and swim freely in water. They can penetrate the skin of the human host during activities like bathing or washing. Human infection. Once inside the human host, Cercaria shed their tails and become Schistosmolia. Schistosmolia migrate via venous circulation to the lungs, then the heart. They develop in the liver and exit via the portal vein system when mature. Adult worm. Male and female adult worms populate and reside in mesenteric venules. Location varies by species. S. japonicum is often found in superior mesenteric veins, while S. memmonsoni occurs in inferior mesenteric veins. Females deposit eggs in venules of the portal system or vesicular and pelvic plexus. As you can see in the picture, the metacercaria insist in the duodenum, then the unembryoned egg is passed out from the species of cow, goat, or sheep, and then embryonic egg. Then Miracidium finds snail host within 24 hours, then eliminates snail host, then circular is shed by snail, finds lives to insist, and then metacercaria, and then the cycle begins. Again repeats. So, trematode life cycle. Ingestion. Sorry, the transmission route. Ingestion. Most helminth infections occur when a person unintentionally ingests helminth eggs. These eggs are passed through the stool of an infected person and can contaminate soil. If a person comes into contact with that soil or food like fruits or vegetables that has touched infected soil, they can contract helminth. Skin penetration. Some helminths can directly penetrate the skin. For example, larvae of certain species can enter the body through the skin when a person comes into contact with contaminated water or soil. This route is common for hookworms such as Necator americanus and Ancyclostoma duodenale. Vector-borne. Certain helminths are transmitted via vectors. These vectors include specific species of lice and mosquitoes. When these vectors bite an infected host, they can transmit helminth larvae to a new host. Now, the transmission of menopause. Ascaris. Ingestion route. Ascaris lumbricoides. Eggs are often found in contaminated soil or food when a person accidentally ingests this egg. Example, through unwashed hands or contaminated produce, they hatch in the intestine and the larva develop. Hookworm. Skin penetration route. Hookworm larvae. Example, Necator americanus and Ancyclostoma rolled in this directly penetrate the skin. Contact with contaminated soil or water. Larva to enter the body through the skin. Leading to people origin. The form of transmitted Uh, sorry, actually my internet connection was weak. Yeah, so we were on pinworm, transmission of nematode. So fecal oral route, the pinworms introduce vermiculars are commonly transmitted via self-inoculation. Eggs near the anus are transferred to the mouth through stretching or touching contaminated surfaces. Ingesting these eggs leads to infection. Transmission of Tinea. Urofecal route Tinea species are transmitted through ingestion of contaminated food or water. Humans become accidental hosts by ingesting tapeworm eggs from contaminated sources. Cross-infection and auto-infection are common, especially in children. Echinococcus, predator-prey route. Now this is interesting. Echinococcus involve a predator-prey relationship. Carnivorous, which are definitive hosts, harbor mature tapeworms in the intestine. Humans, accidental hosts, become infected by ingesting parasite eggs from contaminated food and water. Larval stages developed in the viscera. Transmission route of trematode. Schistosoma. Skin penetration route. Schistosomasis occurs when larval forms of schistosoma parasites released by freshwater snails penetrate the skin during the contact with infested water. People suffering from schistosomiasis can contaminate fresh water sources with feces or urine containing parasite eggs, which then hatch in water. Fasciola. Ingestion route. Fasciola hepatica and Fasciola gigantica are large liver flukes primarily found in domestic and wild ruminants. Humans become infected by ingesting metacercia, contaminated vegetation such as watercress. After ingestion, the metacercia exists in the duodenum and penetrates through the intestinal wall into the peritoneal cavity. The immature flukes then migrate through the liver parenchyma into biliary ducts when they mature into adult flukes and produce eggs. So now let's go to the internal anatomy. The digestive system of the helminth is responsible for breaking down food, releasing nutrients, and absorbing them into the body. Key organs include the alimentary canal, which consists of the mouth, esophagus, stomach, small intestine, and large intestine. Accessory digestive organs like the liver, gallbladder, and pancreas also play a vital role in digestion. Now, let's go to the reproductive system. Helminths exhibit diverse reproductive strategies. Most helminths are dioecious, meaning they have separate male and female individuals. Reproductive organs include testes in males and ovaries in females. Fertilization occurs externally or internally, depending on the species. Now, let's go to the nervous system. The nervous system coordinates various functions in humans. It includes the enteric nervous system, intrinsic innervation, and the ANS, which is autonomic nervous system, extrinsic innervation. Now, plexuses control movement, feeding, and other processes. Now, let's go to the excretory system. Helminths eliminate waste through the excretory system. This system includes specialized cells and structures such as flame cells, protein or excretory tubules. These structures help regulate osmotic balance and remove metabolic waste products. So, as you can see, there is a internal anatomy of the helminth, which you can easily see the intestine, oviduct, pseudocele, cuticle, excretory duct, uterus, muscles, ovary, the mouth, pharynx, excretory pore, intestine, parietal pore, uterus, ovary, anus. So this is the basic structure of the helminth. Now, let's jump to the digestive system of nematodes. Nematodes have a cylindrical body shape that possess a complete digestive system which includes a mouth, esophagus, intestine, and anus. Nutrient absorption occurs through the intestinal wall. Cestodes, which are tapeworms, have a long, flat body composed of segments called proglottids. Interestingly, they lack a digestive system altogether. Instead, they absorb nutrients directly from their host intestine, which is interesting. Trematodes, which are flukes, are flat, leaf-shaped, and unsegmented. They have a simple digestive system extending from the mouth to the anus. Nutrient absorption occurs from the body surface. Reproductive system of this nematode. So, dioecious males have one testis, seminal vesicle, and vas deferens. Females have one or two ovaries, seminal receptacles, uteri, oviductor, and vulva. Eggs laid some parthogenous. Test note. Hermaphroditic males have testes, accessory glands, serous. Females have ovaries, ciliated oviduct, elongated uterus, common genital pore. Trematode. Mostly hermaphroditic, varied male systems with ducts and and multiple hosts. Now let's jump to the nervous system. Nematodes or the roundworm. The nervous system is simple. Enteric nervous system, nerve plexuses along the alimentary canal, controls feeding, movement, digestion. ANS coordinates involuntary processes include sensory and motor neurons. Centralization lacks centralized brain. Testodes or tapeworm. Nervous system simpler than nematodes. Nerve ganglia scattered throughout body controls basic functions. Centralization lacks two brain or cephalic ganglia. Decentralized nervous system. Very similar to nematodes. Trematodes or flukes have more complex nervous system. Nerve ganglia and plexus is located in various regions. Control movement, feeding, reproductive behavior. Sensory structures include sensory papillae, chemoreceptors, detect environmental cues, adaptation, adapt to host environment. Excretory system. Nematodes or roundworms. Nematodes do not have a specialized excretory system. Nitrogenous waste are removed by diffusion through their body. In marine nematodes, specialized glands regulate water and salt balance by removing unwanted ions while maintaining internal body fluid concentration. Testodes or tapeworms. Testodes lack a digestive system altogether. Their excretion primarily occurs through the tegument or body surface. Although they lack specialized excretory structures, some testodes excrete waste through flame cells, proteinephridia, located in the troglodytes. Trematodes or flukes possess an excretory system mainly concerned with osmoregulation. The system includes proteinephridia, which opens into a collecting duct on each side of the body. Nitrogenous waste is mostly excreted through the tegument. Trematodes have complex life cycles involving at least two hosts, an intermediate host, usually a snail, and a definitive host, a vertebrate. The last slide, prevention and control. Hygiene practices. Hand hygiene. Proper hand washing after using the toilet is crucial. Encourage through thorough hand washing with soap and water to prevent contamination. Personal hygiene. Promote good personal hygiene practices such as keeping nails short, avoiding nail biting, and maintaining clean clothing. Footwear. Encourage regular use of shoes to prevent soil transmitted infections. Safe food handling. Educate about safe food preparation, storage, and consumption to minimize the risk of ingesting helminth eggs. Medication. Deworming. Regular deworming with safe and effective medicines such as albendazole or mebendazole helps eliminate existing infections. Preventive preventive chemotherapy. Large scale distribution of anti-helminth drugs periodically can reduce the overall burden of helminth infection. Environmental control. Improved sanitation. Enhance sanitation facilities to reduce soil contamination with effective helminths. Safe water supply. Ensure access to safe and clean water to prevent ingestion of contaminated water. Health education. Educate communities about helminth transmission routes and preventive measures. Environmental services. Regularly clean and disinfect healthcare environments and equipment to prevent transmission. Laundry and bedding. Properly dispose of contaminated and soiled linen, change linen frequently and follow infection control protocols. Animals. Address animal reservoirs and the role in helminth transmission. Regulated medical waste. Handle medical waste appropriately to prevent contamination. I hope you all will follow this prevention and control. Thank you, folks.
So, th- so